I'm Dr. Rajan. So we'll talk about the comprehensive diagnostic tests that are available for tuberculosis as of now. So it's a very important topic uh, that is there for the exams also, and these tests are very important for the day-to-day daily life also as a doctor. So we'll review the diagnostic modalities for tuberculosis. We'll understand the stepwise workflow for each test, and we'll discuss the performance, how to interpret, and what are the limitations of the currently available tests. So if we look at an overview of the diagnostic modalities, so we have microbiological, immunological, biomarkers, histopathology, radiology, and there are some newer experimental tests that are still under evaluation. So microbiological, you can have smears, you can have cultures, you can have CBNAT, you can have LPAs, you can have whole genome sequencing. Immunologicals, amongst others, you have tuberculosis skin test, IGRA, SCI-TB, the newer one. Biomarkers, you have EDA, interferon gamma, and LAM. Histopathology, you can do a biopsy, FNAC, immunohistochemistry using anti-MPT64. Radiologically, and you have the clinical radiological suspicion based on X-rays, CTs, MRIs, and other modalities. And the experimental are the metabolomics, the microbiological load assays, MBLAs, and AI is also coming up in diagnosis. So what are the basic specimen collection principles? So for respiratory samples, we need a sputum sample. So ideally, a sputum sample of more than equal to 3 ml is considered good. You can induce sputum using hypertonic saline and nebulization, and you can go for the bronchoalveolar lavage, other samples also. In extrapulmonary, you can do CSF, pleural fluid, lymph node aspirates, gastric aspirates, especially in children who are unable to cough out secretions, tissue samples, urine and stool also. They are used rarely, but they are there. Again, timing early morning is preferred, especially for respiratory samples, especially sputum. The concept behind that they say is that when you're sleeping, the sample good bacterial load gets collected, and the early morning when you cough it out, the chances of you getting a positive test result is very high. Then in samples in which you have to do delayed tests, like you have to send somewhere, the lab is not near you, especially you have to do molecular tests, you have to maintain a cold chain of 4 degrees Celsius. And you have to do aliquoting, that is, you have to do separate sample collections or separate bottles for smear, culture, CBNAT, and histology. And more for the lab. So if you are a lab which is doing a routine processing of the sample in the biosafety level 2, and where there are steps in which you are generating aerosol, the biosafety level 3, BSL3 is required. So how do you start? First, you have to do decontamination and concentration of the sample. So NaLC, NaOH method, that is the most commonly used method. That is equal volume of NaLC, NaOH is used in the sample. You expose the sample for 15 minutes and then you centrifuge it for 15 minutes at 3000 G. So what is the purpose of decontamination is to kill the contaminating flora while preserving the MTB viability. Then the sediment, split sediment is there. You can use it for culture and other molecular tests. Then you have to label and document the sample with an ID, date, time, and the type of sample you have collected. Quality assurance, you have to have negative and positive controls for each batch. So now coming to the principles. So smear microscopy principles. So as we know that the Mycobacterium tuberculosis is an acid-fast bacilli, it is detected by the mycolic acid staining technique. So there is a ZN staining method called the Zeal-Nielsen heat method, in which you basically stain with carbonyl filtration, you decolorize with acid alcohol, and then you counter stain with methylene blue. Then there are other staining methods also, that is the auramine O or the auramine rhodamine method, that is the light emitting diode fluorescent microscopy. It is faster and higher sensitivity. So we'll go into the details of the smear process in a while. So how do you prepare a smear? So you prepare a thin smear on a glass slide. You either air dry it or you heat fix it, and then you do the staining. Either you do the ZN stain or you do the auramine rhodamine stain. Then you examine the said slide under the microscope and you record the result as per the IUALT, ATLD and the WHO grade guideline, and better is to report it on the same day. So how do you do the staining? This is the ZN stain method. You apply the primary stain of carbon for 30 seconds. Then you heat fix the slide using flame. Then you wash it off. Then you decolorize with acid alcohol for 15 to 20 seconds. And then you apply a counter stain with methylene blue for 30 seconds. And then there is the excess stain. And then you can see the slide under the microscope under oil. So you have in this method, you have the background of bluish with a pinkish bacilli you can see visible. That is the acid-fast bacilli. So what are the smear grading guidelines? So you have to initially see 100 fields. More than that, you can also see. So you label the sample as scanned. If there are no AFB viable visible, then you label it as none, nil, negative. If there are 1 to 9 AFBs per 100 fields, then you label it as scanty. If you find 10 to 99 AFBs per 100 field, then you label it as 1 plus. If there are 1 to 10 AFB per field, so previously it was per 100 field, now 2 and 3 are per field. So if there are 1 to 10 AFB per field, it is 2 plus grading. And if there are more than 10 AFB, then it is per field, then it is 3 plus. Then TB is not the only acid-fast organism. There are other acid-fast organisms also available, are present. So we divide them into three. So that is the true acid-fast organism, partially acid-fast organism, and modified acid-fast protozoa. So in true acid-fast, we have Mycobacterium tuberculosis complex, non-tuberculous mycobacteria. In this, we use the ZN stain or the auramine rhodamine stain, and you decolorize with either acid, alcohol, or acid you can use. Then partially acid-fast or weakly is Nocardia species, Rhodococcus species, Gordonia, Sucamurella, Corynebacterium, and then you use the modified ZN staining, the Phyte Farraco staining it is called, and modified acid-fast protozoa also there, Cryptosporidium, Cystospora, Cyclospora, and again you can use the modified staining methods. So these are some of the acid-fast organisms. There are many more acid-fast organisms that are present. Now coming to the second staining method, that is the auramine rhodamine or the auramine O staining. In this, the fluorochrome dye auramine rhodamine is used to stain the organism. So auramine rhodamine combines with the mycolic acid on the cell wall, which is then fixed with steamed heat. So then you use a decolorizing agent, acid alcohol is used to rinse the non-stained dyes. Then you counter stain with potassium permanganate to stain the non-fluorescent tissue and cell debris. Then you visualize this under the ultraviolet light or the fluorescent microscope. So any principle of any staining generally you use is you stain, then you decolorize, and then you counter stain. So in auramine rhodamine, you will see a background of black with a fluorescent yellowish, greenish, and sometimes oranges mycobacteria can be visible. Then what is the advantages of auramine rhodamine staining? It is a rapid technique as compared to Ziel-Nielsen. It is more sensitive than Ziel-Nielsen, and it does not require heat fixation. You need to steam fix, but you do not need heat fixation for dyes. And the number of fields you have to observe is a little bit less. So it is a little bit less time consuming. But there are some limitations of auramine rhodamine. You have to have a confirmative culture method for this staining, and some of the stains may not appear fluorescent. And if it is negative, then you have to confirm it with the ZN stain method or another method to confirm that it is negative. Then these dyes are possibly carcinogenic, and the contents used are sometimes irritant to the skin and the eyes and the respiratory tract. So if staining technique is not correct and there is excessive exposure to counter stain, so the brilliance of the fluorescence organism can decrease, and you have to observe this stain slide within 24 hours of staining, because otherwise the fluorescence fades away. Now, coming to the culture media, originally they used to use the solid media called the Lowenstein-Jensen or the LG medium. It is an egg-based medium with glycerol and malachite green. So the malachite green gives it the classic green color of the media. So you incubate it at 37 degrees up to 8 weeks. The colony morphology is, it's a buff, rough, and a dry colony. Advantages is it's an inexpensive test. If there is contamination in the sample, that is easily visible. But limitations are that it is slow, and as compared to the newer liquid culture, it has a lower sensitivity. So this is the LG medium on the right side, tube slanting medium you have to put, and then the colony as described as rough, buff, and dry. Now coming to the standardized media, that is the media that is most commonly used, called as the MGIT 960. So MGIT 960 is an oxygen sensor based test which detects the bacillary growth. We inoculate 0.5 ml of the processed specimen into the MGIT tube, and it is an automated, and time to positivity is generally 7 to 14 days, but it takes time sometimes. And the contamination rate is 5 to 8%. Advantages, it is faster, more sensitive, and on this you can even add on automated drug sensitivity testing. So now coming to the MGIT, a little bit of detail. So MGIT is the Mycobacterium growth indicator tube. So it contains the modified Middlebrook 7H9 broth, which is, it's an enriched liquid medium. It contains OADC enrichment, that is oleic acid, albumin, dextrose, and catalase. There is a antibiotic PA panta antibiotic mixture, that is polymyxin B, amphotericin B, naldixic acid, trimethoprim, acetylocillin, to inhibit the contaminants. The bottom of the each tube contains an oxygen quenched fluorescent compound which is embedded in the silicone. As the mycobacteria grow and consume the oxygen, the fluorescence increases, which is visible, and which also instrument also detects it. So now you might be asking, why is it called as 960? So 960 denotes the automated instrument can accommodate 960 cultures tubes at a time. So the instrument of the MGIT can accommodate up to 960 culture bottles. That's why it's called 960. So it looks like this. So Mycobacterium growth indicator tube MGIT, samples in ultraviolet light, emission of this orangish fluorescent indicates the presence of mycobacteria. Now you can even put phenotypic drug susceptibility testing on this MGIT tube. So it can detect the first line drugs. It can detect the second line drugs also, and injectables also. And the method used is called as proportion method, and it takes around 1 to 3 weeks for drug susceptibility testing. And you have to follow the guidelines by the WHO 2021 technical manual guidelines are available, which says how do you say this drug is susceptible or not. So what is the stepwise procedure for this? First, you prepare the inoculum, a standard suspension of 0.5 McFarland of the empty acid from a positive MGIT or LG. So you have a positive MGIT or LG culture, preferably the MGIT. You take standard suspension. Then for how do you set up the tube? For each test, one control MGIT tube in which you do not put the drug, and one or more drug containing MGIT tubes are inoculated. So you use this to inoculate one tube which has no drug in it, and one or more tubes which has the drug. Then you place these tubes in the MGIT 960 instrument, and you monitor the fluorescence for every 60 minutes. Now, how do you interpret? When the control tube reaches a predefined fluorescent threshold, that is, growth control is equal to 400 growth units, the instrument then compares automatically growth in the drug-containing tube. So when the control tube reaches 400 growth unit, then the instrument automatically compares it with the drug-containing MGIT tube. So If the growth in the drug tube is more than equal to 100 growth units, then you say that the organism MTB is resistant to that drug. If the growth is less than 100 units, then you say it is a susceptible MTB. Now coming to the cartridge-based nucleic acid test, which we commonly use, CBNAT, that is the cartridge-based nucleic acid amplification testing, called as the expert MTB with rifampicin resistance. So what is the principle behind this? The principle is it's a real-time PCR. It detects MTB complex plus RPOB mutation, that is a mutation for rifampicin resistance. So how do you prepare the sample? You mix 1:2 sputum with sample reagent, then you incubate, and then you put it in the cartridge and you put it inside the CBNAT machine. So generally, the time for turnaround time is 2 hours. For smear positive, the sensitivity is 95%, smear negative is 60 to 70%, specificity is more than 98% for both smear positive and negative. So what are the steps? You can see first sputum liquefaction and inactivation with sample reagent. Then you transfer 2 ml of the material into the cartridge. Then cartridge is inserted into MTF RIF test platform. Now here the hands-on work ends. Now everything else is automated. Fourth step is sample automatically filtered and washed. Then fifth step is ultrasonic lysis of filter captured organism to release the DNA. Then the DNA molecules are mixed with dry PCR reagents. Then semi-nested real-time amplification and detection in integrated reaction tubes, and then you can get a printable results. Now there is something called as Expert Ultra, which has a higher sensitivity. So in normal MTB, CBNAT, you detect 131 colony forming units per ml in Expert, while in Ultra you can even detect as low as 16 colony forming units per ml. So in Ultra, even trace results are present. It is generally useful for pediatric TB, T meningitis, and EPTB. Now coming to GeneXpert MTB XDR, that is the comprehensive TB and drug resistant detection. So it can go beyond rifampicin resistance. It can detect other resistance also. So it's a platform by Cepheid. It's a US based company. It detects rapidly MTB and the multi-drug resistance. So it detects the mutation in CATG, INH, GYRA, GYRB, and the RRS. So additional detection capabilities along with rifampicin is the isoniazid, ethionamide, fluoroquinolone, injectables, amikacin, carnomycin, capreomycin. So now TrueNAT platform. So it's an Indian platform, and there are some places in Delhi which have started using TrueNAT. It's a chip-based PCR for MTB detection plus rifampicin resistance. It's portable. It is battery operated for peripheral labs. And stepwise workflow is similar, that you prepare the sample via lysis and extraction, then you amplify, and then you detect. So sensitivity is 89 to 95% and specificity is 98%. So it's a small portable kit, TrueNAT, and it's an Indian based. Now, if you compare the other TCBNAT platforms, so there is something called as GeneXpert Omni that is also available. So it's smaller, it's portable, and the other things are same only. That it takes TB and rifampicin resistance. It is highly portable. The time is around 2 hours, and WHO ICMR approves it. Then now there is a test called as a PathoDetect, which is similar, but it detects additional INH resistance also. Then, as I said, GeneXpert Ultra, TrueNAT, Expert Edge, Express. Again, these are portable and rapid working, and XDR. Now, something called as TB LAMP, loop amplification method. It's an isothermal amplification, and turbidity fluorescent readout is there. It is used as a microscopy replacement in peripheral labs. So if you don't have hands in the periphery, or you don't have microscope or able technicians, then you can use this. Sensitivity is 77 to 80% and specificity is 96 to 98%. Limitation is there is no drug resistance detection. So what do you do? First, you do a sample preparation. You collect 60 microliters using ICAN disposable pipette in the sputum cup. You put it in the heating tube. Then you put it in the machine. So that's the DNA extraction. You shake it and you put it there. Again, the principle is fluorescent based only. So fluorescence is there, then the TB is detected. If it's not, then it is not detected. Now coming to the important topic, that is the line probe assays. So these are basically newer tests which are there, which can help in for genotypic testing of the resistant pattern. So genotype, there are two: genotype MTB DR plus, that is for the rifampicin isoniazid resistance, and genotype MTB DRSL fluoroquinolone plus injectable resistance. So the principle is similar to CVNAT. First we do a PCR, then we do an hybridization, then you have to do a band pattern interpretation. So direct testing of smear positive specimen or culture isolates for smear negatives. But there are newer generation LPA which are not readily available in India, but they can be used even in smear negative also. But the limitation is only known mutations are detected, and heteroresistance may cause mixed bands, so difficulty in interpretation. So what is the principle of this line probe assay? So LP are a family of DNA strip-based tests that allows user to determine the drug resistance profile of an MTB complex strain by interpreting a pattern of band that represent lines in immobilized probes that are found to MTB amplicons. LPA probes are designed to target the most common mutation associated with resistance to first and second line anti-TB drugs, and there are specific MTB complex file type DNA sequences called as the WT DNA sequences. Mutations are detected by binding the amplicons to probes that target the most common mutation, that is the MUT probes, that is the first and second line LPA, or by lack of amplicon binding, that is lack of hybridization to the corresponding WT probe. I'll go into detail in the next few slides. How do you actually interpret it? So this is the configuration of the genotype MTB-DR plus version 2 and G in the left side, and the DRSL3 in the right side. So genotype MTB-DR plus targets specific mutation in the RIF resistance determining region of the RPOB gene from the codons 505 to 533 to detect RIF resistance and mutation in INH promoter from -16 to -18 nucleotide upstream, and CAT-G. So basically, the first one, if you look at the A, there is something in the line 1, there is something the conjugate control, then there is something called as amplification control. Then the third line is the M tuberculosis complex TUB, and then there are different probes. So from 4 to 16, you can see RPOB, locus control, then fifth is wild type. So these are all linings, which I, again, in the next corresponding slides, I will explain. So the second version of the genotype includes quinolone resistance determining region using GYRA and GYRB, fluoroquinolone and RRS, and EIS promoter region for resistance to the amikacin. Now, if you look, the second line here is called as the amplification control. So what is amplification control? So the AC band verifies the successful completion of the DNA amplification step. So basically, the funda of AC amplification control is, okay, it says that the polymerase chain reaction or the PCR has been completed successfully. So it contains a synthetic DNA fragment that is amplified along the patient's DNA. The appearance of the AC band confirms that the PCR process worked correctly and was not inhibited. So if the AC band is present, it indicates that the DNA extraction and amplification step was successful. For you to say that the test is a valid test, the amplification control line should be present and should be visible. If the AC band is negative, it indicates that the test is invalid. This can be caused because of problems with amplification process, such as reagent issues or the presence of inhibitors in the patient sample, and the test must be repeated. If the AC band is weak, sometimes the weak band can occur in a test with positive result. This can happen due to competition for reagent during the application process. But if the other bands are clearly visible, then the test is still considered valid. Now, the first line is the conjugate control. So, conjugate control band verifies the successful completion of hybridization and detection steps. So, amplification says that the PCR has been completed successfully, while the conjugate control says hybridization and detection steps have been successfully completed. So, it ensures that the enzyme labeled conjugate is active and correctly binds the detection system on the test strip. This confirms that the chromogenic reaction, which produces the visible band, has occurred correctly. So CC band is present, confirms that the proper function of conjugate and chromogenic reaction, and this should be present for a test to be considered valid. If it is negative, then it indicates that the test is invalid. This can be due to faulty conjugate, expired reagents, or an error in the hybridization or color development process, and hence the test should be repeated. Now, how do you interpret and report? So LPA has two internal controls on the strip, as I explained, the conjugate control and the amplification control. The conjugate control line should always be visible in order to document the efficiency of conjugate binding, and the amplification control serves as a reference for interpretation of WT and MUT probes. Only those bands of which the intensity is about as strong or stronger than that of amplification should be considered. So in the WT and the MUT probe, which I'll explain in the following slides, only those should be considered if they are as strong as the amplification control or stronger than them. So in the first line LPA, when there are strong signals in the WT bands but weak or no staining in the amplification control band, that is a single WT band that is significantly fainter than the remaining WT band of signals should be considered negative. So again, as I said, key amplification is no staining or weak, so you should not consider that as a valid test. In case of a negative test result, both the conjugate and the amplification control band should be visible to ensure that a valid negative result. So basic underlying is that you should always ensure that the amplification control band and the conjugate control band both are positive. Now, how do you interpret the resistance pattern? So there are two types of probes. One is the wild type probe, that is the WT probes, and the mutation probes, the MUT probes. So in wild type probes, so these probes are designed to bind to the DNA sequences of the drug susceptible version of the target. So these are probes which are designed to bind to the DNA sequences of the drug susceptible version of the test. So if the WT band is present, it indicates that the susceptible DNA sequence is present in the sample. So that means that the sample is sensitive to that drug. Now, mutation probes. So these probes are designed to bind to the mutated DNA sequence that are known to confer drug resistance. So if the MUT band is present, then it indicates that the resistance conferring mutation is present. So WT probes are present, it means that it is sensitive to that. And if the MUT probes are present, it indicates that it is resistant to that. So how do they work together? If it's a drug susceptible result, if the all relevant WT bands are present but no MUT bands are present, this means that the bacteria is wild type and it is susceptible to the drug. While in a drug resistant result, if the WT band is absent and a corresponding MUT band is present, it indicates that the presence of drug resistant mutation is present. So these are four case scenarios. What can happen? So case one is all WT probes are developed and all MUT probes are not developed. That is, resistance is not detected, it is sensitive. Then one or more WT probes are not developed and one or more MUT probes of the corresponding regions are developed. Again, now this is that it is a resistant, again depends on the drug. Resistance detected. Then one or more WT probes are developed, not developed, but no MUT probes are developed. It again depends on resistance. And one can be there that all WT probes are developed and one MUT probe is developed, that is resistance detected. And you have to go to the identification sequence. So this is how you see the LPA. So if you look, first is the CC line, that is the conjugate control. Second is the amplification control, and third is the TB complex, that is the TB is present, MTB gene is present for detecting of the organism. So all these three should be positive, and then you can consider it valid and positive. So this is how you label. So if you look in the first one, it is CC is present, AC is present, and TUB is present. And then if you look, the wild types are present and MUTs are absent. So this is a sensitive sample. So this is how you read the sample. Now coming to the immunological test for tuberculosis. So the original tuberculin skin test or the Montu test. So what is the principle behind it? It measures the delayed type 4 hypersensitivity reaction to purified protein derivative, PPD, which we inject intradermally. The procedure is that 0.5 ml of 5 TU of PPD is injected intradermally on the forearm, and induration is measured at 40 to 72 hours using a transparent ruler. So you have to record the induration in mm, not the arrhythmia. You record the arrhythmia, but the induration is the one that is matters. Sensitivity is in immunocompetent host 70 to 90% sensitive. In HIV, mandatory and elderly sensitivity is lower. Specificity is poor in BCG vaccinated or NTM exposure. Again, limitation is false positive in BCG, NTM, false negative in immunosuppression, recent TB infection, viral illness, requires patient to return for reading, and no restriction between latent and active TB. Now, something called as interferon gamma release assay, IGRA. So, its principle is based on quantiferon, to quantify the interferon gamma released by the T lymphocyte sensitized to MTB antigens. Specific antigens are ESAT6, CFP10, and TB7.7. These antigens are absent in BCG and most NTM, thus they have a higher specificity. So you have quantiferon TB Gold Plus, which uses the whole blood ELISA, and T-Spot TB, which is the ELISpot platform. Now, quantiferon TB Gold is blood directly drawn into 4 tubes. One is the nil control, one is TB1, CD4 plus, TB2, CD4 plus, and CD8 plus, and mitogen, that is a positive control. You incubate it for 16 to 24 hours, and then the quantify the plasma interferon gamma level by ELISA, and the result is expressed in international units per ml. How do you interpret? If the interferon gamma is more than 0.35 IU per ml after backslash substitution, it is indeterminate if it's nil, or nil is more than 8, or mitogen less than 0.5. So sensitivity is around 80 to 90%, specificity is more than 95% better than TST, and it retains accuracy in BCG vaccinated populations. Limitations are it is expensive, requires ELISA facilities, and has a limited role in high burden active TB settings. And it again cannot distinguish between active and latent TB. Now, second is T-Spot TB test. So it is incubated with MTM antigen. So this uses ESAT6 and CFP10, and interpreted more than 6 spots on above nail is positive, and it is sensitive in immunocompromised hosts also. Slightly higher sensitivity than quantiferon Q15 HIV in children, and more robust to immunosuppression. Now, there is a newer test called as a TB test. That's a cytokine-based T cell assay. So it's a developed in Denmark and also in Serum Institute in India. So it's a CTB test. It again detects the ESAT6 CFP-based skin test. So it combines the simplicity of a tuberculosis skin test with the specificity of the IGRA. So mechanism is same as IGRA. Recombinant ESAT6 and CFP injected intradermally like PPDD measures local delayed hypersensitivity response mediated by sensitized T cells, and read again at 48 to 72 hours, similar to Montu. So interpretation is induration more than equal to 5 mm is considered positivity. Again, there is no BCG cross reactivity. Again, field friendly, high specificity sensitivity, unaffected by BCG, comparable sensitivity to IGRA, and useful for mass latent TB infection screening in high burden areas and BCG populated vaccinated populations. So this is the basic difference between TST, IGRA, and SCI-TB. TST, you use PPD. IGRA, you use ESAT6, CFP10, TB 7.7, while in CTB, you use ESAT6 and CFP10. So it's a TST is a skin test, IGRA is a blood assay, SCI-TB is a skin test. BCG interference is there in TST, not there in IGRA, and not there in SCI-TB. Infrastructure requirement is minimal in TST SCI-TB, while you require an ELISA lab in IGRA. Sensitivity 70 to 90%, 80 to 90%, 85 to 90% respectively. Specificity 50 to 70 in TST, more than 95, more than 95 in SCI-TB and IGRA. Cost is TST is low cost, IGRA is highly expensive, and SCI-TB lies somewhere in between. So all three can detect latent TB, but unfortunately, three of them cannot differentiate the latent from the active TB infection. Now come to the last topic, that is what is the whole genome sequencing. So whole genome sequencing determines the complete DNA sequence of the Mycobacterium tuberculosis. It identifies all mutations, including those associated with drug resistance, virulence, and strain type. So what are the four main key applications of this? So first is the drug resistant detection. So detects mutation for first line and second line drugs. It can identify resistance before the phenotypic methods, and it is very helpful in epidemiology and outbreak tracking. So molecular typing for strain clustering, transmission mapping, and outbreak investigation is helpful. Then you can use it for phylogenetic analysis, determine the lineage, sublineage for MTB for epidemiological studies, and it can also detect mixed infections, detects multiple strains in a single patient sample. So you collect the sample, sputum, culture isolate, or other clinical specimen. You do a high quality genomic DNA extraction from MTB. Then you do a library preparation, DNA fragmentation, and adapter ligation is done. Then you use the platform, that is NGS platform, next gen sequencing platforms, and then you report the comprehensive resistance and epidemiological data. So the advantages of the whole genome sequencing is detects all known resistance mutation in a single test, faster than phenotypic drug susceptibility testing for multiple drugs, enables high resolution epidemiology for TB control programs, and also identifies novel mutation associated with drug resistance. What are the limitations and challenges? It requires high quality DNA, usually from culture isolates, need advanced library, laboratory infrastructure, and trained personnel. Data interpretation requires robust bioinformatics, and it is costly and not yet point of care in most settings. So what is the current role and guidelines for WGS? So WHO recommends can be used for MDR and XDR detection in reference laboratories, supporting surveillance and outbreak investigations. In India, the National TB Elimination Program (NTEP) has limited use in tertiary labs for drug resistance and surveillance. So thank you so much.
Thank you, Dr. Rajan, for a wonderful talk. Now I request Dr. Ankit to please continue.
Thank you, Dr. Rajan, for a wonderful, insightful talk. And it is the most common thing we are seeing day by day basis. And I just have some questions in mind, and I myself clear to all of you who are joining. As we are practicing last 15 to 20 years, uh, almost no day is gone without seeing TB patient. Whether you do bronchoscopy or not, you do sputum, you do biopsy, or you do any tissue, uh, always a patient is there in your OPD, IPD who came out to TB positive. So the first thing in my mind is that can you diagnose on clinical or radiological basis that it is a MDR or DRTB patient? No. Actually, on the clinical basis and radiologically, X-ray and CT scan, you can label it as that it may be tuberculosis. But if still you are not having microbiological analysis or you are not having pathological analysis, then you could not label it as a tuberculosis patient. Sometimes we used to see that in around 15-20 years back that there was a primary MDR in the family, and all family members later on, after 3 months, 6 months, or 10 months, came out to be primary MDR positive on GeneXpert or LPA. So this may be there, and sometimes you should start ATT empirically for 3 weeks, and you should see clinical response, radiological response, or you do sputum if available, or you may do tissue biopsy, or you may take out ball, bronchial lavage. So the another question in my mind is that if a patient came to an OPD and sputum came out to be scanty or 1+, so can you start ATT? As we are living in the highly prevalent uh, tuberculosis and endemic country, India. So you see there are a lot of patient. Those came out to be sputum positivity as trace or 1+. So these has to be correlate clinically as well as radiologically. You do not start upfront ATT without confirming clinically or radiologically. Sometimes we used to see around 1 in 1,000 or 1 in 100 that no clinical radiology criteria match and you have to start ATT. But that is exception. But if the sputum is scanty or 1+, you have to reconfirm from the microbiologist, or you have to talk to them. You may do another sputum sample for AFB, or you may another consider nocardiosis or histoplasmosis or other causes of the sputum scanty causes such as false positive, such as nocardia, actinomycosis. So you have to see vigilantly the patient at his or her reports. Do not directly go for the ATT in a patient who came out sputum scanty or only 1+. So the another question in my mind is there is something histoplasmosis and nocardiosis. As if sputum sample is 1+ or scanty, there may be also dead vessel and there may be also contaminant. And sometimes we used to see around 2-3 cases of histoplasmosis in a year. That is very, very difficult for a microbiologist to identify, differentiate from tuberculosis. As I am working in a private setup from last 10, 12 years, and there is cost also. So it is difficult to do all the tests. In government setup, it is slightly easier to do other tests. So if you have very much doubt on TB or histoplasmosis, always do tissue biopsy, either through bronchoscopically guided or lymph node biopsy through the surgeon, or you may expert in true cut biopsy from the lymph node, or you have some intervention radiologist who is expert in taking tissue diagnosis. And it is the pathologist who may diagnose histoplasmosis. Microbiologist is rare to see to diagnose histoplasmonocardiosis because it is difficult to obtain sample from the patient every time. And second, there is a you have to do modified Jaden stain or Gimori stain or PAS stain to differentiate between the three. So please consider histoplasmosis or nocardiosis in all your patients who are immunocompromised, who are having or advanced ILD, who are having bronchiectasis, who are having previous history of tuberculosis. Do not jump directly to the tuberculosis if the GeneXpert or other reports are highly positive. Then another thing is how many samples we have to take. We have to tell the patient to go for the sputum for acid fast bacilli testing. Actually, SBAT started around 30-40 years ago, and it was in the Tomen, that is the Bible of tuberculosis, that was released by the WHO, that you have to give 3 early morning sample or either 2 early morning sample or 1 sample after the lunch. And it was seen that if you give 2 early morning sample of 1 in early morning sample or second sample after the lunch or after the breakfast, some hour of breakfast, it may have same result, same sensitivity, that is 3 sample. So it is the you who has to decide that you have to give 2 sample or 3 sample for the patient benefit. Do not follow blindly the guideline, follow the patient. And the another thing that is commonly missed, the when to use LP, line probe assay, as around 15 17 years ago, I was having DRTB ward, and we have LPA diagnostics, because at that time GeneXpert was started in India in 2010, and we do not have a GeneXpert, so we started LPA in our lab, and result was great. So in the guideline, if you see 2016 TB guideline, if any patient is sputum positive, you should either go for CBNAT, or you should either go for LPA. Or if patient is history of tuberculosis previously, not responding to treatment in 1 or 2 months, or history of contact, or there is extensive tuberculosis, or there is a primary MDR in the family, you can directly go for LPA. But nowadays, GeneXpert is more common. LPA is slightly less available even in the metro cities. And the GeneXpert is better in tissue diagnosis, but in LPA, only the sputum positive and somehow CSF and pleural fluid, they are slightly sensitive. But GeneXpert is best for rifampicin resistance and the tissue diagnosis than LPA. LPA is only for sputum positive, that sample. So you have to see when LPA to be done. And sometimes you see that there are patient sputum reports are negative, and pathologists suddenly say you did some endobronchial biopsy, or you did some TB-LB, and pathologists suddenly say it is looks like tuberculosis. So pathological diagnosis is always considered tuberculosis unless proven otherwise. So you should believe on pathologist, and you should discuss with your pathologist and microbiologist along with your clinical correlation that what to do. Generally, I am doing that from last 10 years, and sometimes I see that they are correct, sometimes I am correct. So you should always do biopsy correlation with your clinical radiological correlation to rule out TB or histoplasma or nocardiosis, many things. And there are labs which are using TrueNAT, as Dr. Rajan has beautifully covered. GeneXpert Ultra, TrueNAT. So TrueNAT is also very sensitive test. If it is there in your city, you can go for TrueNAT also. It is equal to around GeneXpert Ultra. Okay. And the last thing, how samples to be sent on to be sure that which sample is good for TB diagnosis. Actually, sometimes patient is not able to generate sputum, or repeated sputum sampling came out to be negative for TB diagnosis. So you must do BAL, or if there is a EPTB, you can do pleural fluid CSF. Then you should subject to CBNAT and culture, everything for the patient sake. And the tissue diagnosis is sensitivity specificity is more than 95% in GeneXpert CBNAT, but not in the LPA. So tissue is always good, and sometimes you do the BAL and GeneXpert came out to be low, but in follow-up patient is not improving. So you should either do repeat BAL, or you should rule out other causes. Now, the last thing that we are seeing that GeneXpert Ultra, there is a result called trace. So you are doing bronchoscopy for pneumonia, that is patient is history of acute illness, and chest access resistive pneumonia, patient is not improving, you start doing bronchoscopy, and GeneXpert came out to be trace detected. So you have to start it or not. It depends on you how to carry on for that patient. So please go through your clinical, radiological, GeneXpert report, talk to your microbiologist, then you decide. Because I have seen many GeneXpert Ultra are actually catch the dead bacilli, and they are not the actual result. So we have not started the ATT, and these patients were doing well from last 10-15 years. And we move on to questions if anybody has asked. Any other questions you can ask? I am there to answer.
Thank you, sir. There are questions from the audience. First question is, with so many rapid molecular tests available now, do you think microscopy will ever become obsolete in TB diagnostics?
Actually, microscopy is the gold standard in TB diagnosis as well as the GeneXpert. We do not know that it may become obsolete in the era of AI. But nowadays, even I see that the last 2 3 years we are conducting DNB examination, and we are seeing students are not knowing on microscopy how AFB seen and how it looks like, because I think they are not doing by themselves. So I think microscopy is the first thing in the resource limited setting to rule out tuberculosis. And the other things can be, if your resource limited setting has GeneXpert, then it may overcome microscopy in future. But I think microscopy must be there.
Thank you, sir. Next question. If a patient on treatment for MDR TB is not responding clinically, should I consider NGS? What additional information could it provide?
Actually, MDR TB patient, as per the latest treatment protocol, BPLAM regime, we generally do not able to keep BPLAM regime in our hospital. We used to refer to the national hospital like Tuberculosis Hospital, LRS, Lala Ram Sarup Hospital, NITRD, Maroli. But if you are uh, suspecting, you are starting ATT on the previous regime, that is a long regime for the MDR patient, and on around 2 to 3 months or 6 months, you expecting patient is not improving, you may consider repeat testing, and you follow the previous culture report, which is suggestive of tuberculosis or not, or you may see previous DST report. Then you may order another report, or you may rule out another causes, because I have seen patients hide history of smoking, history of diabetes, history of hypertension, history of hypothyroid. So there are some comorbidities that may also lead to decrease in result, and there is seen, there may be seen that patient is not improving, or sometimes the that is vitamin D deficiency and even the other micronutrient deficiency lead to decrease in efficiency of antitubercular drug, and you see that patient is looks like not responding to the treatment. But you have to follow as per the guideline, and if you want to treat, you can treat. I have treated around 15 years ago. Then we used to treat around 9 months to 1.5 year, and around our result were very good because we don't have NITRD lab at that time. But it is up to you and where you practice.
Thank you, sir.
And
Next
lastly,
one question.
and lastly about NGS. Actually, the NGS is a newer test. We still do not use it, but you may use if it is pocket friendly to your patient.
Sir, last question is about the treatment. If BPAL fails due to linezolid after 1 month, how to change treatment? What to do about bedaquiline?
Actually, we have last lecture BPLM regime by Dr. Vicky Bakshi that was beautifully covered by him. And we do not give BPL regime because I am working in a private hospital. So he is the best person to answer. I will forward your question to Dr. Vicky Bakshi, who will answer you, and we will forward to you also. Sorry for that.
Thank you, sir. That were all the questions.
Can we close now?
Yes, sir. Thank you, everyone.
Thank you, sir.